There's a tooth in a laboratory somewhere right now that came out of the ground after 40,000 years. And researchers are pulling DNA out of it. Here's the part that most people don't know. The DNA they're most likely to recover isn't from the nucleus. It's from the mitochondria, which brings me to something I find genuinely strange, and that almost nobody learns in school. You have two genomes. Everyone knows about the first one. 23 pairs of chromosomes, sitting in the nucleus of your cells, half from each parent. That's the one that gets talked about. But inside your mitochondria there's a second, completely separate genome. Its own loop of DNA, physically outside the nucleus, replicating on its own schedule. Let me describe it, because the specifics are interesting. It's small. Roughly 16,570 bases. For comparison, your nuclear genome runs to about 3 billion. It's circular rather than linear. And it's extremely compact, with very little wasted space. It contains 37 genes. And here's where it connects to everything else I talk about on this channel. 13 of those 37 genes code for proteins involved in oxidative phosphorylation. That's the process that uses oxygen to produce ATP, the energy currency your cells spend. In other words, they're building parts of the electron transport chain. The remaining genes code for the transfer RNA and ribosomal RNA that the mitochondrion needs to actually manufacture those proteins locally. So the mitochondrion has its own genes, its own machinery to read them, and it makes some of its own components on site. It's semi-independent in a way no other structure in your cell is. Now, why does any of this matter to archaeology? Two reasons, and both come straight out of the biology. The first is copy number. Your nuclear genes exist in 2 copies per cell. One from each parent. That's it. The mitochondrial genome exists in hundreds to thousands of copies per cell, because you have many mitochondria and each one carries multiple copies of its DNA. Now think about what happens to a body over thousands of years. The DNA fragments. It gets chemically damaged. Most of it becomes unreadable. If you're hunting for a sequence that only ever existed in 2 copies, your odds are terrible. If you're hunting for one that existed in thousands of copies per cell, some of them survive. That's the whole reason mitochondrial DNA dominates ancient DNA work. It's a numbers game, and the mitochondrial genome starts with a huge advantage. The second reason is inheritance. You got all of your mitochondrial DNA from your mother. The egg cell brings mitochondria into the embryo. Sperm contribute nuclear DNA, and essentially nothing else that persists. And there's another feature that matters enormously. Nuclear DNA gets shuffled every generation through recombination, mixing maternal and paternal contributions. Mitochondrial DNA doesn't do that. It passes down intact, mother to child, generation after generation, changing only through the slow accumulation of mutations. That makes it a clean lineage marker. If 2 people share a mitochondrial sequence, they share a maternal line. And by counting the differences between sequences, researchers can estimate how long ago 2 lines split. That's how maternal haplogroups get defined, and how ancient remains get connected to living populations. Now I want to correct something, because if you've been reading about ancient DNA you've almost certainly run into it, and it's almost always described wrongly. Mitochondrial Eve. The way it usually gets presented is that there was a single woman, the mother of all humanity, and we all descend from her. That is not what the finding says. What it actually says is this. If you trace every living person's maternal line backwards, mother to mother to mother, those lines eventually converge on one individual. The technical term is the most recent common ancestor of the mitochondrial genome. Current estimates put that convergence at somewhere around 100 to 200,000 years ago. But she was not the only woman alive at that time. Not remotely. There were many thousands of women in her population. Many of them have descendants living right now. What's special about her is narrow and specific. Only her unbroken female line survived to the present. Other women's maternal lines died out at some point, whenever a generation produced no daughters, even though their nuclear DNA may well have made it down to us through sons. So she's not the first woman and not the only ancestor. She's a bookkeeping point in one particular lineage. And there's an honest limitation researchers themselves point out. Because that convergence is relatively recent in evolutionary terms, mitochondrial DNA has limited power to tell us about the deeper past. It's excellent for the last 100,000 years or so, and it gets weaker beyond that. That's why modern ancient DNA work increasingly tries to recover nuclear genomes too, when preservation allows. Alright. Let's bring this back into your body, and to what you can actually do. You have a genome inside your mitochondria that includes the instructions for 13 components of your energy machinery. Reasonable question. Can you influence it? Here's where I have to give you a genuinely split answer, and I think the split is more interesting than a clean answer would be. There's a study I keep coming back to. Researchers trained 10 healthy untrained men using a one-legged knee extensor protocol for 6 weeks. That design is elegant, because the untrained leg acts as a control inside the same person. Same diet, same sleep, same everything. The results in the trained leg were dramatic. Endurance performance up 120%. Power output up 49%. Peak oxygen uptake up 33%. Citrate synthase activity, a standard marker of mitochondrial content, up 51%. And the activities of respiratory chain complexes 1 through 4 up between 46 and 61%. That's an enormous change in the working capacity of those mitochondria in 6 weeks. Now the surprise. When they measured the amount of mitochondrial DNA, it hadn't changed at all. Neither had the markers for inner and outer mitochondrial membrane. Their conclusion was that these adaptations happen independently of the number of mitochondrial genomes, and probably come from changes in how actively that DNA is being transcribed. Not more copies of the blueprint. More output from the copies you already have. And when they detrained the leg for 4 weeks, the enzyme activities fell back down. Mitochondrial DNA still didn't move. Other researchers have found different results. There's work showing mitochondrial DNA copy number does rise with high-intensity training, correlating with improvements in respiration. So the field hasn't settled this. I'm telling you both sides because you deserve to know which parts are solid. What's solid is the outcome. 6 weeks of training roughly doubled endurance performance and raised respiratory chain enzyme activity by around half. That's not in dispute in that study. The mechanism underneath it, whether you're making more genomes or working the existing ones harder, is still being argued. And notice something. That uncertainty doesn't change what you should do. Either way, the input is the same. So the free lever today is the same one that keeps showing up whenever I look at any of this honestly. Use the muscle. Endurance work in particular. That's the intervention with the strongest human evidence behind mitochondrial capacity, and it's free. Now, where my own recommendation sits, and it's narrow. 13 of those 37 mitochondrial genes build components of the electron transport chain. That's the chain CoQ10 works in, as one of the carriers moving electrons between complexes so ATP can be produced. Levels of it tend to decline as we get older. I want to be precise. CoQ10 is not encoded by your mitochondrial DNA. It's a molecule, not one of those 13 proteins. I'm not claiming any supplement changes your mitochondrial genome, its copy number, or how it's transcribed. Nobody has shown that, and it would be a very easy thing to imply today given the topic. What I'm saying is narrower. The genes are building the assembly line. CoQ10 works on that assembly line. That's the relationship, and nothing more. So move first. That's free, and it's the part with real human evidence. And if you want to support the machinery, the link in the description below is an affiliate link to Mitochondrial, which is PQQ and CoQ10, with a 90-day money-back guarantee. If you want more videos where the popular version of a story gets corrected rather than repeated, subscribe. This video is educational only and it's not medical advice. Talk to your doctor before starting new or more intense exercise.